I would like to ask Peter Krawitz now to take over the floor. So he is head of the Genomic Statistics and Bioinformatics at the University of Bonn. Who didn't that know? So his talk is about TNAMSE, which is an acronym for Advanced Genetic Testing. And also here we will hear about networking, and networking is in these days key for our success. So Peter, please go ahead with your talk.
Yeah, thanks for the kind introduction. So now I'm not able to see the live stream anymore, so I have to ask someone else whether everything works. Can you see?
So you are live. You are in the upper right corner with your video, and the presentation. The presentation is showed in full screen.
Okay, great. So yeah, I have to apologize myself a little bit because I'm basically making myself now a fool because I didn't prepare as well as the others. But this is representative for how much we are lacking behind in genome diagnostics, if you want, in Germany compared to the other nations, partially at least. And the best possibility we have to catch up is probably continuing with TNAMSE, which might be a blueprint for what we can contribute on a European level too. So let me quickly explain to all of those from you who are not yet familiar with TNAMSE what it is. So TNAMSE is a care project. I don't know how else to translate ein Versorgungsprojekt. And it is the result of the National Alliance of Patients with Rare Diseases that formed and is supported by the government. And one urgent need is to speed up the time to diagnosis. So this is clearly the goal of this project, and genetics is at the core of it, or next generation sequencing approaches there too. And the consortium is basically headed by Heiko Krude in Berlin. He's the speaker of this consortium here. He's right now meeting with interdisciplinary board of clinicians. This helps him currently help his glasses, I think, and he will also present the main results of this project in the Rare Disease Day in a couple of months. So who else is in this consortium? It's 9 sites in Germany that contribute data. It's mainly the centers of rare diseases, which are a prerequisite for recruiting patients here for this project. And this might also play a crucial role in the continuation. And it's clearly not the first study that evaluates the cost effectiveness of whole exome sequencing, which was the objective here in this project in a national healthcare system. But nevertheless, even if it has been shown by the Netherlands, by Australians, that it is cost effective, you have to show it again in your own healthcare system. And there are differences in the German system that make it unique. It's a multi-payer system, and in this case only BARMA and AOK were participating in this innovation fund funded project. I don't want to go into too much detail here. Most of us are familiar with the peculiarities of the reimbursement. It's often silly, it sounds often silly, and I think the reason for that is that the progress is so quick that everything that you write down is outdated after a few years. So even though maybe the 25 kilobases threshold might have made sense some years ago, it seems ridiculous right now. So I think it's better to think about it as a low cost and high cost NGS test, and whatever is advanced is probably what we should think of as the high cost genetic test. Anyhow, it's not my job in this consortium to present those results, and that's why I would like to put you off for the Rare Disease Day, where Heiko Krude will announce the official results of this project. But what I can do today is go a little bit through the literature, what has been done by other groups, how do we compare to those results. And many of you will be familiar with the results from Stark et al., who did it basically for the healthcare system in Australia. It was a comparable setup, so they also wanted to see is it more cost effective to do whole exome sequencing basically as a first test. And this is indeed what they found out. Also, the numbers are actually not that different. So it's comparable to the reimbursement scheme here. And is exome still relevant, or is that already outdated? Well, partially yes. If we look into more up-to-date publications from the UK, they talk now about whole genome sequencing of patients with rare diseases. But now basically you will experience that I prepared rather a lecture than a presentation, because I want to point out some parts in the papers here. So what did they actually find out? So here I plotted their findings with respect to recent genes. That's just 95 in a cohort of what was it, more than 13,000 individuals. And think about the unconfirmed cases here as candidates. Think about it maybe with a p-value in mind, similar to what Christian Gillison presented earlier. So 95 recent diagnostic grade genes and roughly 19 candidates doesn't sound that much anymore compared to the size of the cohort, and also the data that they generated. So there's yet another figure I would like to point out, and that is how the findings distribute over diagnostic disease genes. So it's roughly exactly 329 diagnostic grade genes that they reported. The top 3 featured in a quarter of all reports, and 19 diagnostic disease genes in a further quarter. So honestly, they could have done 50% with those silly disease genes that we just want to get rid of, right? So I don't want to pledge for panels now. I'd rather emphasize how important it is to define the inclusion criteria for the expensive diagnostic test, right? And that's where the Translate NAMSE consortium did a pretty good job. So another paper, the OrphanNet database, or the OrphanNet group, looked at the prevalence of the rare diseases. We all have in mind this definition of a prevalence below 1 in 2,000 to qualify for a rare disease, but actually there are orders of magnitudes of rareness below this threshold. So it really makes sense to talk about rare diseases and ultra rare diseases. And ultra rare diseases represent the majority by number of those rare diseases, which is visualized basically here. So more than 90% are ultra rare. And now we really need to catch up, and can only solve that by the same approach that the Dutch people basically excel in, or also the UK. And TNAMSE might be a start. So let's look at our numbers. It's enrollment in 9 centers. All of them have these multidisciplinary teams that can assess whether an expensive genetic test makes sense. Exome sequencing has been selected for 1,539 patients, a small number compared to what I reported from the UK, right? And still, the total amount of DDGs that we reported is much larger, right? That compares to 300 something. The diagnostic yield is comparable if we go to these diagnostic subgroups. And here on the left is basically the distribution. How many reports were on 1 gene, or how many of those DDGs have just been reported once, and so on. So the distribution fits much more to what has been shown by OrphanNet, right? Translate NAMSE is excellent for the ultra rare diseases. The others have already been solved. And now let's also look at the timing. 79 of the genes that we reported, we could only report because the phenotypes have been published within the last 5 years, right? And if you normalize that per year, it's roughly 1% of diagnostic yield that you gain just by reanalyzing the data. This is in line with stuff that has been published by others, by Wright et al. And again, also the candidates that we found. All those candidates were a result of sharing data. And the importance doesn't need to be emphasized anymore, but how do we achieve that? It's obviously important to use protocols that allow us to exchange data beyond borders. And the Beacon protocol is probably the best way to go. We can integrate those APIs into the software that we are using for analyzing the data. And by that means, we will be able to link our data and join basically the other large consortia and projects that are already operative. And everyone who would like to join in that effort, I think TNAMSE or whatever comes after will be open to many more centers in Germany, can join our developers round. I just wrote down the software frameworks that are in use in this consortium right now. Barfish, GSWA, EVADB. We will update it on the TNAMSE website, and we will be happy if you want to join this developers round to get maybe even another software ready for data sharing. That's the point. Okay, now some more numbers. So I already mentioned the diagnostic yield. It differs for the major disease groups. It also differs for kids versus adults. It's also very enjoyable for me right now to communicate with all the other geneticists about their opinions on differences I couldn't explain myself, for example. So maybe you also wonder about the delta in between kids and adults for immunological diseases. Most likely the explanation is that this subgroup of disorders is most life-threatening. So maybe there are just less adults with severe immunological born diseases. Maybe they are less severe because they are less monogenic. Might be one explanation in the adults. Anyhow, we can also look at the distribution of AD, autosomal dominant versus autosomal recessive causes. And here we basically confirm what's known for 10 years now, the shift of paradigm for ID. It's mainly due to de novo mutations, at least in our community, or at least in the part of our community that has a low relationship coefficient. And this is also work I like quite a lot from Martin et al., where they showed at the proportion of dominant versus recessive in different mating schemes. So it's the same environment, it's all the UK, but EABI is of European ancestry, and that's more likely to be random mating, and PABI is from Pakistani ancestry, so that's more likely to be a consanguineous partnership, represented here with higher autozygosity. And I find this plot here the most challenging part for interpretation is, I think, the part that's missing here in the EABI, which is unexplained. So we can confirm that also in TNAMSE, the proportion that's solvable by recessive disorders is much higher, around 30%. It's maybe a little bit easier to find a homozygous mutation that's disease-causing in a recessive mode of inheritance. But I don't think this big chunk here of unsolved cases are due to compound heterozygous or non-coding, which are tough to detect. There is a hypothesis which I have and would like to investigate further, in this draft here, we simulated prevalence and mutation load and showed that the lower prevalence in a random mating population is actually just a transient phenomenon after growth phase. If you are interested in the simulations, I would be happy to link a clip here for you too later on. But right now, I'd like to draw your attention basically to the higher mutation load. So the higher mutation load is basically the disadvantage of this drop in prevalence, which is just transient, right? You achieve it by an increasing mutation load. So in a larger random mating population, the mutation load should be higher, and maybe we actually should go more for oligogenic or degenic oligogenic modes of inheritance to explain this missing part of heritability. Again, something that we can only do with interconnected databases. Okay, now, second part, we had the chance to pigtail or jump on this Translate NAMSE project also with research studies. And we published already a proof of concept paper about the value of computer-assisted image analysis or pattern recognition. And now we had the chance to test our performance in a prospective study. So the workflow is sketched here. We include also portrait photos if the patients consent to that use in the workup of the exome data. And again, I'd like to give you an update on our algorithms now, because I explained to you already that we need to deal with the ultra-rare disorders in TNAMSE or whatever follows after. So I like to show those three pictures here usually as a starter, but in this audience, it's probably pretty straightforward to name the first one. It's Kabuki makeup syndrome. Obviously, I'm a fan of Marbury syndrome, which some of you might also know, but very few will get the last one right. So how is that? Why is that? It's because the prevalences are so different. So the human experts in the end have the same problem as the machine, which currently supports just the more frequent ones, right? So everything that's supported by DeepGestalt is rather frequent, and now I show you how we gonna tackle the problem with the ultra rare ones. So I skip over that quickly. Many of you know DeepGestalt within Phasogene, so these are the syndromes that you will get, and this is not yet supported. Some 101 on deep learning. So the problem here is that we are dealing with rare data sets. The variation is large. The number of classes is unknown. We try to solve it with knowledge transfer. It's pretty good with portrait images because we can learn from the open domain what a face is. This has been published. It's something that's available in an app. And now let's deal with the rare ones. So here we start with the same architecture. We train the network with everything that's known, but now the magic happens. We use the feature layer for the definition of clinical phase phenotype space. And in this clinical phase phenotype space, we are now able not only to match known disorders, which DeepGestalt already supports, but we are also able to match the unseen syndromes, unseen for the system, right? And this is the first disorder where we succeeded to describe this high similarity. It's actually the same de novo mutation. And I find it so revealing that the artificial intelligence actually agrees with the human experts on distinctiveness. So here on the left-hand side, you basically see the syndrome distinctiveness as assessed by syndromologists, Nadja Emke, Denise Horn, Karen Grip, Shahida Mosa, just to name a few of them. And it correlates with the accuracy that the AI achieves. And it does not correlate with the prevalence, right? That's what you see on the right-hand side. And the highly distinctive one with number 1 is, I think, PAX1, which Christian also mentioned in his talk already. So it's available now to beta users. It's living within the phase2gene environment, but we certainly aim for an open source project here. So if you want to use it as a clinician, it should be available right now. Contact us. And basically, the workflow is representing how clinicians work. First, you try to solve what's known. But if you don't succeed, then you are also interested in the matching approach, right? Then you might want to contact a clinician that has a similar case. Okay. And again, this is what many of you know. We added with the PDA workflow the support for image analysis, and we can do that with Gestalt Matcher now too. So this is the original vector machine that has been trained. Data curation was an important part of it. That was all from the proof of concept. And now, as I already mentioned, it's validated in TNAMSE. We can use that approach to prioritize the disease genes. It's doing a pretty good job. Again, for diagnostics, it's always time that matters too. We have to increase the diagnostic yield, but we also have to achieve that in a decent time. And this is basically represented by the top 10 accuracy rate or top 1 accuracy rate. And now in the last 5 minutes, let's think a little bit about what's ahead. So exome was TNAMSE, or TNAMSE was mainly exome. And Genome DE will be more than that. I think it's not yet clear what Genome DE will be. Except that it needs to be a meaningful contribution to what's going on the European level. I think also we shouldn't restrict it to genomes. It's not all we need to solve it. There is proteome. There's also obviously transcriptome as the link. And just as an anecdote before coming to an end, one example where genomes might actually help, or which cases might actually be solved by a genome, although there are so many variants that are hard to interpret. And I take a Kansel 1 case, or a Kohlen de Vries patient, so took that from their website. It is often due to microdeletions, but also single nucleotide variants or small indels have been described as causative. But not all those patients that might have the suspected diagnosis of colon de Vries can be solved by exome sequencing. And here basically I show you why, because many of the small deletions which you don't detect in an array and which are mainly in the intron and not deleting an entire exon are missed by exome sequencing, right? But on the other hand, if you have a phenotype score that is so indicative for this syndrome, which is indicated here by this high Gestalt score in the lower right, then you need to look at the region in the genome that fits to that phenotype. And then it is actually straightforward to interpret those many variants because it's not many anymore. You restricted your search. You restricted your search space by phenotype information. So if that's possible, I think genome is a way to go. And I'd like to end now. I'd like to thank mainly Zhong Xian, who is the PhD student working on Gestalt Matcher. But for everything in Translate NAMSE, there are too many names. I just want to show again the ZSEs of all the people that contributed to TNAMSE. Okay, thanks a lot.
Peter, thank you very much for this exciting talk and the insight. I do have one question. You mentioned that you have currently different mutations, 364 diagnostic grade genes. Who is going to make the decision then to add new DGGs to the panel?
No, sorry. I have to make clear that this is not a panel. It's just a grade that you need to achieve to be of diagnostic relevance. So it's a term used in the Turo et al. Paper to differentiate between a gene for which you can write a report right away or a gene that's still a candidate. And maybe my joke was bad that half of the UK data could have been analyzed by panels. It was just a bad joke. You need exomes. In Translate NAMSE, you need the exomes.
Okay, so that depends on the phenotypic association then of those genes, if you look into that, right?
Yeah, exactly. It's you can also think about it as genes that have been described in omims with phenotypes.
Yeah, yeah. If there are new genes reported on, who's going to? Are you analyzing then retrospective all the data again to ensure that probably the detection rate is getting up or more reproducible?
Yes, exactly. So basically the idea of TNAMSE was to share data within this consortium, and the centers actually started doing that just quite recently. So that's also why I told you that this bioinformatics developers round is kind of work in progress. Many of the candidate genes still have been identified by the very old-fashioned approach, by writing emails, calling the other lab, do you have another case with mutations in this or that gene, right? But it's very inefficient. So that's why the key objective for this developers round is basically to integrate these beacon queries so that this is done more efficiently by IT.
Okay. Another question is, how do you secure that the tools you are developing get into the right hands? You talked about medical professionals are going to get that, but it's always a possibility to copy that and to give it also to, or hand it out to people who have not the right skills to do the interpretation. How do you secure this?
Okay, I think that's also why we positioned this talk in this session after ITSG, right? We need to collaborate with the professionals here. And still, maybe this concept of decentralization is actually an advantage here too. So the beacon protocols do escalate there's an escalation principle for exchanging sensitive information. So you will only be able to exchange that information on a higher security level, right? So maybe you start with the mutation in the beginning, but as soon as it comes to exchanging portrait photos, you really want to be sure whom you are talking to.
Okay. Thank you, Peter. And I'm sure you're open to questions also during the day or on